let's discuss the last question of this paper, that is question number 6. Figure 6.1 is a simplified diagram representing a section through the human immunodeficiency virus HIV particle that causes HIV or AIDS. The diagram shows the virus particle about to attach to the cell surface membrane of the T helper cell at a receptor protein called CD4. A second protein co-receptor called CCR5 is also necessary for the virus particle to enter and then infect the T helper cell. So guys, over here we can see that HIV virus can infect T helper cell, and HIV virus, in order to infect the T helper cell, the HIV virus enters the T helper cell. For the entrance of the HIV virus into the T helper cell, CD4 receptor protein is required and CCR5 co-receptor protein is required. So what will happen is that HIV will bind to the CDR4 receptor protein on the T helper cell, and then this co-receptor, which is CCR5, will help the virus to enter the T helper cell by endocytosis. Alright, part A, identify structure X in figure 6.1. So if we look at the structure X, X is basically a structure which is surrounding the viral nucleic acids. We know that in a virus particle, the nucleic acid is surrounded by the protein coat, which is known as capsid. So the answer is capsid. Structure X is capsid, which is the protein coat. Part B, explain how the ability of the immune system to resist the damaging effects of pathogen is affected by destruction of T helper cells. So guys, over here they are asking that what would happen to the immune system or its ability to destroy the pathogens if the HIV virus causes the destruction of T helper cells. You all know that HIV infects the T helper cell and destroys it. This results in the fall in the number of T helper cells. So you all know that T helper cells secrete cytokines. If T helper cells are being destroyed, then few cytokines will be released. Few cytokines will be secreted by the T helper cells, or we can just write that few cytokines will be released. And because of this, what will happen? Less B lymphocytes will be stimulated, right? Less B lymphocytes or fewer B lymphocytes will be stimulated to differentiate into plasma cells. So fewer plasma cells will be there and fewer antibodies will be produced, right? And less macrophages will be activated, so less phagocytosis. All right, so fewer cytokines will be secreted, less stimulation of B lymphocytes, less stimulation of B lymphocytes, and hence less clonal expansion, and less plasma cells, or we can say fewer plasma cells. Fewer antibodies secreted. Fewer antibodies are secreted, right? We can also say that less activation of or less stimulation of macrophages. So fewer macrophages activated. Fewer macrophages are activated, right? You can also talk about T killer lymphocytes. Fewer T killer lymphocytes are stimulated. As you all know that the cytokines from the T helper lymphocytes stimulate the other cells of the immune system, and with few cytokines being released, the other cells of the immune system will not be fully stimulated, there will be lower stimulation. So fewer T killer lymphocytes are stimulated. Alright, let's move on to part C. Studies have shown that some individuals did not become infected with HIV even though they were repeatedly exposed to the virus. Later discoveries indicated that these individuals had a mutation in the gene for the CCR5 co-receptor protein. Suggest how the mutation of the gene for the CCR5 co-receptor protein provided protection against the HIV infection. So guys, over here they are saying that there were some individuals who did not develop AIDS or HIV even when they were repeatedly exposed to the virus, and this was because of a mutation in the gene which was coding for the CCR5 co-receptor protein. As mentioned earlier, that this protein is required for the virus to enter the T helper cell. So if there's a gene mutation in CCR5, so CCR5 co-receptor will be abnormal, and this will prevent the endocytosis or the entry of HIV into the T helper cell. So we can simply write that suggest how the mutation of the gene for CCR5 co-receptor protein provided protection against the HIV infection. We can simply say that virus cannot enter the cell, or HIV, virus or HIV cannot enter T helper cell because of abnormal CCR5 co-receptor. Co-receptor protein. Alright, let's move on to part D. The use of monoclonal antibodies against the CCR5 co-receptor protein, anti-CCR5, has been shown to be effective in the treatment of HIV infection. Outline how anti-CCR5 monoclonal antibodies can be synthesized in the laboratory using the hybridoma method. So guys, this is a very basic question in which you just have to tell the examiner that how monoclonal antibodies are synthesized in the laboratory using hybridoma method. So you all know that firstly an antigen is injected, an antigen which in this case is CCR5. Is injected into a small mammal, such as a mouse. Then immune response occurs over several weeks. Immune response occurs, and you all know that immune response takes several weeks. Then what do? We extract plasma cells from the spleen. You all know that whenever an antigen is injected, you all know that whenever an antigen is injected into a mammal, what happens? Immune response occurs against that antigen, and B lymphocytes are stimulated, right? And B lymphocytes specialize into plasma cells, and plasma cells secrete antibodies. So what we want to do is that we want to extract the plasma cells which are producing the antibody against CCR5, that is anti-CCR5. So what we do, we just extract all the plasma cells from the spleen first, and then we fuse the plasma cells with myeloma cells. That is the blood cancer cell. Fuse the plasma cells with myeloma cells to form hybridoma cells. To form hybridoma cells. All right. And then we carry out screening to separate desired hybridoma cells which are producing desired monoclonal antibodies, desired monoclonal antibodies which are actually CC, which are actually anti-CCR5. Anti-CCR5. So guys, what is the purpose of the fusion of plasma cells with myeloma cells? Plasma cells cannot divide. However, myeloma cells can divide. So when we fuse plasma cells and myeloma cells, a hybridoma cell is formed which can secrete antibodies like plasma cell and which can also divide like myeloma cell. So we want the cell division so that a lot of cells can be produced and that they can produce a lot of antibodies. They can produce a lot of monoclonal antibodies, right? And why did we collect all the plasma cells from the spleen? Because we exactly don't know that which one is our desired plasma cell. So we take out all the plasma cells from the spleen, we fuse the plasma cells with the myeloma cells to form hybridoma cells, and then we screen for our desired hybridoma cells. The desired hybridoma cells are those hybridoma cells which are secreting our desired monoclonal antibodies. So guys, we are done with this paper. Thank you so much for watching. Allah Hafiz everyone. Don't forget to subscribe to my YouTube channel.